Welcome to my second session on Drug Development Process Part 2 prepared by Ruhul Emin. Drug screening involves a sequence of iterative experimentation and characterization. A variety of biologic assays at the molecular, cellular, organ system, and whole animal levels are used to define the activity and selectivity of the drug. The type and number of initial screening tests depend on the pharmacologic and therapeutic goal of the lead molecule. Mechanism of action and selectivity of the drug play important role in drug screening. There are good predictive preclinical models exist, example as antibacterials, hypertension or thrombotic disease. We generally have adequate drugs. Good drugs or breakthrough improvements are conspicuously lacking and slow for diseases for which preclinical models are poor, or not yet available, example Alzheimer's disease. The desired result of this screening procedure is called a lead compound, that means, a leading candidate for a successful new drug. A patent application would generally be filed for a novel compound that is efficacious, or for a new and non-obvious therapeutic use. Here is some examples of molecular screening models. Here is some examples of molecular screening models. Here is some examples of cellular and disease models used in new drug development process. Drug characterization. Drug characterization involves finding of biological and psychochemical properties of potential drug molecule. Biological characterization. Interaction of drug molecule with biologic systems. Chemical characterization. Physical and chemical characteristics of a potential drug. Biological characterization. Biological characterization is needed to assess whether the molecule will likely work in humans. Goals of biological characterization. 1. To find efficacy. 2. Assess toxic effects. Studied at the level of biochemical, cell, tissue, organ and organism levels. Biochemical assays includes mechanism of action at a molecular level. Receptor binding, affinity and selectivity, assays. Enzyme inhibition activity, Km, selectivity. Metabolic processing metabolic activation. Cellular assays includes cell cultures, cell penetration, receptor activity, transport. Isolated tissues, effects on specific tissue, possible toxicity. Liver tissues, microsomes drug metabolism, drug interaction. System/whole animals models includes rat, mouse, toxicity, LD50, organ-specific efficacy. Dog, rabbit, guinea pig, monkey, pharmacokinetics/pharmacokinetics, BA, toxicity. Lab animal models for disease, efficacy, toxicity. Gene array chips, gene expression. Chemical characterization in LUDs. Physical and chemical properties of a drug are critical for knowing how it can be formulated/stored/administered. Chemical structure, mass spectroscopy, NMR, X-ray crystallography. Solubility, pK. Partition coefficient, octanol water. Stability. Impurities, TLC, HPLC, GC, mass spectroscopy.